welcome to my new channel, Revisionary NEET, the ultimate pathway to NEET triumph. So we are starting with the topic Kingdom Monera of the chapter Biological Classification. We are going to revise the whole topic. So let's begin. So in Kingdom Monera, we learn about prokaryotes, all of the prokaryotes. They are actually bacteria who are the sole members here. They are the most abundant in nature, occurred in almost every place. We can find a handful of soil full of bacteria. So they are likely most everywhere in the earth. They also are found in extreme habitats like deserts, snow, deep oceans, hot spring, etc. Bacteria structure is very simple, but they are complex in their behavior. So the extensive metabolical diversity are shown in bacteria. Bacteria can be divided in four groups. They are bacillus, which are rod-shaped, coccus, which are spherical, vibrio, which are comma-shaped, spirillum, which are spiral-shaped. Bacillus is the most common one here, and coccus can be non-motile. Here the gram staining topics are mainly out of NCERT, but if you want to read, you can read. I will share if you need. Then we will learn about the classification of Monera. Monera are divided into two main parts. They are eubacteria and archaebacteria. We will learn about the archaebacteria later. And eubacteria are mainly, there are two characters, that they have rigid cell wall, and if motile, they have flagella present in their body. Eubacteria are further divided into three parts: bacteria, cyanobacteria, and mycoplasma. We will discuss it later. Details. First, we will learn about eubacteria. Their nutrition in bacteria is divided into two parts: autotrophic and heterotrophic. Autotrophic means they can produce their foods of their own. Heterotrophic means they can't. So in heterotrophic bacteria, they are also divided in three parts. They are saprophytic, parasitic, and symbiotic. Saprophytic means they gather their food from dead organic matters. Parasitic means they are pathogens and harmful to us and other animal spirits. Their examples are Vibrio cholera, Clostridium tetani, Solanarum typhi, and Xanthonas citrei. Symbiotic bacteria can be nitrogen fixers, mainly are nitrogen fixtures, and they also can be free living in some cases. Their examples are Rhizobium frankia, Escherichia coli. So we have learned about heterotrophic bacteria. Now they are autotrophic bacteria. Autotrophic bacteria are divided into two parts: photoautotrophic and chemoautotrophic. Photoautotrophic bacteria use sun energy, and chemoautotrophic bacteria use chemical energy. Chemoautotrophs plays a great role in recycling nutrients. They oxidize inorganic nutrients such as NH3, nitrite, nitrate, etc. Chemoautotropic bacteria can be sulfur bacteria, iron bacteria, nitrifying bacteria, hydrogen bacteria, etc. Nitrifying bacteria, they are important, like Nitrosomas, Nitrococcus. In the photoautotropic bacteria, there are anoxygenic photoautotrophs and oxygenic photoautotrophs. The examples of oxygenic photoautotrophs are blue-green algae, means cyanobacteria. Blue-green algae or cyanobacteria are mainly nitrogen fixtures, and they can be examples of Anamina, Nostoc, Oscillatoria, Olocera. Heterotrophic bacteria are also helpful in human affairs, like nitrogen fixtures in plants. They turn milk into curd, which lactobacillus bacteria helps here, antibiotic producing, etc. Respiration in bacteria is out of NCERT, not totally given, but if you can read, you can pause and study it. Bacteria can reproduce by three methods: asexual reproduction, true sexual reproduction, and short of sexual reproduction. True sexual reproduction is absent in them. It's important. They asexually reproduce by binary fission and by producing endospores. In NCERT, short of sexual reproduction, they are meaning that these can be conjugation, transformation, transduction. We will learn in class 12 chapters. Here comes the most important parts of the chapters. There's cyanobacteria or blue-green algae. They are oxygenic photoautotrophs, and they are autotrophic photosynthetic bacteria released free O2 in atmosphere as the first organisms. They have chlorophyll like green plants, but they are not. So there's important point. They have phycobilin pigments, the C-phycobilin, C-phycoerythrin. They causes red color of Red Sea. But don't confuse it with red tide. Red tide will learn in dinoflagellates. That will come later. They can be unicellular, colonial, filamentous. Filamentous example are Nostoganavina, Oscillatoria, Spirulina. In filamentous bacteria, the colony of filament always surrounded by gelatinous or mucilaginous sheath. They can be occurred in freshwater, marine water, terrestrial. Blue-green algae often form algal bloom, which which is because of dissolved O2 in water, which is harmful for aquatic life. They can reproduce by binary fission and fragmentation. They are mainly nitrogen fishes that I told you earlier. They have some important characteristics like gelatinous sheath, cell wall is made of peptidoglycan, plasma membrane, cytoplasm, nucleus is single, circular and double-stranded. 70S ribosome they have. They have gas vacuoles, which are also found in purple sulfur bacteria and green sulfur bacteria. They also have lamellasomes. They are important characteristics of them, that semicircular structure attached to plasma membrane. They have chromatophores, which are membranous extension of them, which contains pigments like PS1 and PS2. In nostoc, we can see some vegetative cells of them, but one cell is different there. That's called heterocyst. Important characteristic of nostoc, the specialized cells for nitrogen fixing. Then we will learn another type of bacteria, which are mycoplasma or PPLO. That's what called we pleuropneumonial like organism that are first found in cattle's lungs pneumonia infection in their pleural fluid. That's one of the most important points here, that they completely lack cell wall. So their shape is not fixed in time. So that's called pleuromorphic. They are the smallest living cell here. They can survive without O2, so they are called facultative anaerobics. They are heterotrophic in their nutrition. Many of them are pathogens. They can pass through the bacteria-proof filter. Now we have learned all the heterobacteria. Now are going to learn the archaebacteria. They can also live in some harsh conditions. Their difference is in the cell wall structure. Archaebacteria cell wall is made of pseudomembrane, so that's why they are different. Archaebacteria are mainly divided in three parts: methanogens, thermoacidophiles, and halophiles. Halophiles, which they can live in salty areas, so they are facultative anaerobe and heterotroph. Thermo means temperature, and so they can live in high temperature and high acidity. They can live in hot spring. They also are facultative anaerobes. Methanogens live in marshy areas, in gut of ruminant animals like cow, buffalo. Then from their dung, we can produce biogas, which is important. They are obligate anaerobes and chemoautotrophs. So that's all for my first video of this channel. Do let me know if the video was helpful. There are too much mistakes in the video, but I will like to improve and suggest some improvement if you have any. So like the video, subscribe the channel for daily contents. Signing off. Thank you for watching.